Test, test. Good afternoon, everyone. Just so you know, lunch is served. So on the right-hand side, we have the regular people, and the vegetarians are over on the left-hand side, where you'll also find the drinks. Please help yourself. Good afternoon, everybody. In about 10 minutes, we're going to be starting, so if you can grab some food, find a chair, and there's quite a few people here, so get cozy, and if you're got something on the chair next to you, please put it under the chair so people can sit down. So in about 10 minutes. Can we get everybody to take some seats so we can start the presentations? That'd be great. Vendors, can you stop talking for a little while so people can migrate towards the seats? Grab some food. So welcome everyone, my name's Tom Pfeifer. I'm going to be your host for this afternoon, and we've got a great lineup of speakers and vendors around for you to participate in. Hope you've all had a chance to grab some food, grab a drink or something. They'll be here for the rest of the time. We'll have a short coffee break afterwards as you get to mingle with the vendors. But yeah, welcome to Toronto Spring Event 2024 for LRIG Canada. It's our second in a row. Our first one was last year in Montreal. Before we get started, I just do a land acknowledgement. We acknowledge we're on the traditional territory of many nations, including the Mississauga, the Credit, the Anishinaabe, Chippewa, the Haudenosaunee Confederacy, the Wendat peoples, and home to many First Nations, Inuit, Métis people. Vendors in the back there, can I get you to participate here, please? Thank you. So, Robotics Innovation Group. We're a not-for-profit. I don't know what you know about the society. You got to start in the early 80s when they were basically using tank turret to turn around and move test tubes around and smashing them up a lot. There's about 15,000 people worldwide in LRIG, various things. A lot of them in the US, obviously, but it's growing out in Europe, Great Britain, various other places in the world. And hey, we got to start one here in Canada, and that's what we're kind of proud of. So our Canadian chapter members, LRIG was incorporated in 2022. We held our first Montreal event last year. And I'd just like to acknowledge the founding members. I don't know where they are, but if they could just kind of step forward and stand up. Elizabeth is currently at SLS Europe, so you won't see her around today. We've got Ross somewhere. There he is over there. Simon is out front along with Xavier, who's managing people walking inside here, myself. And the five of us got together and decided we should try to do something like this in Canada. We also have a number of volunteers, and if they could just kind of step forward here, we've got Andres somewhere at the thermo booth there, Bammy, who's unfortunately not here because she's on maternity leave, busy, and Joyce is around here too, and they've helped us put all this thing together. It takes a lot of people to drive and do these kind of things. We're always looking for volunteers. It's useful. It's helpful for people to get involved if you want to make some new connections around the country, around the world. So please participate. There's lots of little places where you can pick up the QR code, sign up as LRIG, because after all, we're here to help you guys out, look at automation and things more. So our mission statement really is we're a nonprofit. We're just trying to fund these kind of things so that people come by, have a lunch, chat with some vendors, and get some great information about stuff going around the country. We empower communication with scientists, engineers, academics, and we try to foster self-development, collaboration, innovation, knowledge sharing, and advancing the field of laboratory automation, okay? It's a mouthful, but basically we're here to help you figure out what robot you need to do whatever you need to do or how you can build something, which is the other side of things. So like I mentioned earlier, join us. It's free. Become a volunteer, and even just join us within the LinkedIn group. We do most of our communication through our LinkedIn groups. That's how we communicate. It's the easiest thing for us to do for most of the things there. So these things are around in various places and tables. Please grab them. Grab the QR code from the screen or wherever you need to, or come and talk to us. We're happy to put you in touch with various things. A big thank you to all our sponsors. Take a look around at you. All these people that have sponsored this event, made it happen. Please take the time to spend 5 minutes with each one of them, see what they've got, see what they can teach you about various things. Without the sponsors, this event would not happen. Okay, so please take the time during our coffee social hour aspect of it and say hi to them. So we've got a great event here from you. We are going to be starting off with Robin, so a little genomics. you know, a little, what we're going to call, sorry, just lost the word there, but a little translational thing within stem cells, and then we're going to end it off by a little bit about chemistry at the very end. Okay, we're very excited about it. Please stay till the very end because it'll be very interesting to see how you apply automation things to chemistry, which can also apply to a lot of the life science words. So without further ado, I'd like to introduce to you Robin, and Robin Coope is coming from us from the BC Genome Science Center out in Vancouver, British Columbia. He got his PhD in physics and gravitated towards automation a little bit by talking with a lot of clinical scientists. He's head of the project groups within the BC Cancer Agency, where he helps create all the robots and everything doing this thing. He's been involved in a lot of company startups, made a lot of cool devices. You can talk to him about that at some other time. Here he's going to tell us a little bit about how Genome Sciences has solved some of their automation issues over the last few years. So Robin, please.
Thank you. Thanks for coming. I feel grateful that I am able to run a fully formed laboratory automation group in a Canadian biomedical institution. It's really cool and we have a lot of fun. I just wanted to actually kind of describe my team just to sort of give you a picture of the kind of skill sets involved. So Dwayne there in the black t-shirt does full-time Hamilton robot programming. And this actually turns out to be very important, just programming all day and not sort of faffing about with meetings and other nonsense that the rest of us do. And that is actually quite important in this whole process of really studying what you're doing and learning. He's supported on your left by Steve Pleasance, who is we call him the god of Excel sometimes. He's kind of a data master, also does Janus programming, and also writes low-level executables to make the Hamilton robots do custom things that Dwayne then uses. And then we have Angus Wong there next to me, who is a full-on mechatronics engineer, brilliant guy, one of a line of mechatronics or various types of actual engineers who worked for me, who all left me eventually to go off and do wonderful things, and they're all still my friends, which is great. In terms of the robots, we have 9 Nimbus 96s, the smaller Hamilton robot. They have 12 deck locations. Remember, we're primarily doing human genome, so we're doing smaller numbers of samples where each well in the plate is like several thousands of dollars of work. So we haven't had to sort of do big scale-up things with plates, but we're trying to automate relatively smaller assays. We have a couple of starlets. The one in the back there is, in that picture on the right, is a biosafety cabinet from DFMZ. If that was a condo in Vancouver, you could move into it. It's so big, and it would cost you about $750,000. The actual one, it wasn't it wasn't that expensive. Anyway, then we have a couple of Januses that we use at the front end in biospecimens and back end for some pooling and stuff. Lower volume, decently accurate at the low volumes. The big model we have is working with our colleagues in the technology development team, Yongjun Zhao's group, so primarily Simon Hiley and Pavan Pando, who are either developing novel methods or adapting methods commercially. And then the idea is to actually really write that down as a very clear method with labware and volumes and reagent and what is going where, and then they get together with Dwayne, and so there's 2 people going to be looking at this thing, and they program and they iterate, and they decide if they like it or not, or they change it. And just having those 2 eyes and those 2 separate skill sets working on these things seems to work really well in terms of delivering a good product. I like to imagine that genomic library construction is generally on the harder end of a lot of library or a lot of liquid handling. I don't know if that's really true. We've certainly been challenged by much of it. The red boxes there are various kinds of purifications for which we use magnetic beads. I'll talk about that in a sec. They involve a lot of recovering all of the sample, so no dead volume left in the wells. That's hard. Obviously, there's some quant setups, which everybody does, which are low volume. And then there's some tricky reagents, again, the alcohols for the purifications, and some very expensive and slightly gooey enzymes, which need to be handled carefully. Really, a lot of it's about bead cleans. And so if you haven't encountered solid phase reversible immobilization or spry beads, they're styrene beads with magnetic cores. They look like kind of like not very well made coffee. You put DNA in it, and with the alcohol, DNA doesn't like alcohol, it sticks to the beads. You then put the beads, you put the well in a presence of a magnet, and they remarkably all clear off. And if you have the correct shaped magnet, you get a donut. And then you stick your pipette tip down the middle of the donut with the clear liquid. Now, all of the beads have gone away. And then you suck out the stuff, and you can then wash the beads with more reagent. If you then put an aqueous reagent in and take it off the magnet, everything resuspends, the DNA floats off, and then you can pull the beads away again and then get the DNA away from the beads, and you're done, and it's brilliant. And no more centrifugation or any of this nonsense. We've also discovered a trick where sometimes, depending on your plate geometry, you want to actually pull your beads down to help resuspend in small volume. And so we were messing around, there's a point to this later, but we were messing around with little spacers of moving the plate higher to pull the beads down farther, depending on what we're doing. I'll get to that in a bit. In terms of the structure, we're obviously a big operation. We've been doing this for a long time. We have a heck of a lot of methods. And this would all be an absolute nightmare to manage, except a long time ago, and this is really Martin Hurst, who's now director of Michael Smith Labs at UBC, who's a professor out there, is really we should, it's all genomic sample prep. We should harmonize to the greatest extent possible the modules within all the things we're doing. We're doing enzyme setups, enzymatic reaction setups, and we're doing bead purifications. And so we've worked very hard to harmonize many protocols. And so in the end, across DNA and RNA and lots of things, we are ending up using essentially one bead method with some volume variations here and there. And that just makes our code structure overall vastly easier to manage. And it just has avoided just sort of drowning in different codes sitting on different robots and the sort of things that large, well-established labs have. The other thing we do then is we have all methods on a SVN server, and we actually, and there's a bunch of technical reasons for this, we push out a new copy every 24 hours to all the robots. So all the robots have all the code on them, like all the Nimbuses have all the Nimbus code on them, and they can run, in principle, any one robot can run any of the code, although we actually have RNA extraction, and then we have pre-PCR, and then we have post-PCR, but they're all basically compatible. And again, that just makes kind of administration of the whole place easier. Again, as I said, with Steve Pleasance, we can write bespoke robot functions. So that means we can use the lower level libraries, and we can write executables where you call the executable from the Hamilton method, and then the robot does a bunch of cool stuff. And I'll talk more about some examples of that, and then come back to the end of it. And as long as your pipe tips are empty, then you can kind of go on and using regular method things. So we can do sort of aspirate mix things, and we can also change the translation speed of the head and so forth. How did we get there? So way back, this is an early project, automated size selection. We got involved in Cancer Genome Atlas project, major genomics, and really profoundly successful project actually starting in about 2009. My leadership came back and said, remember that thing you claimed you could make a kind of size selection robot? We just agreed to do all the microRNA for the Cancer Genome Atlas project. So that thing you said you could build, it now needs to work. And so that was essentially the problem of microRNA separation, where the microRNAs are like 22 bases long, and then you have like 88 bases of adapters. And so the difference between the construct, adapter, RNA, adapter is not much different from the adapter-adapter dimer, which is the great enemy in Illumina type sequencing. And so we made a hole-to-hole size selection. So you have a bunch of entry holes up at the top there. This is a plate of 8 lanes. We have 12 of these on the robot deck. We separate, we have individual control of the speed of separation on each one of the wells. Wait till the mRNA arrives. You target it with a camera. It was actually at the back end of that. It's somewhere in there, is like we identify where the microRNA was. Gigantic linker dimer peak you want to get rid of, and you could suck it out. So we ended up doing about 11,000 microRNAs, and further about 20,000 samples on this thing, or on this. Originally, this robot, this is actually a hand-drawn version from my former engineer, Jared Slobodin, who helped spin this thing off, or went with the spin-off company. So this was all built out of scrap robot parts. That was actually from a Qpix. This is an old Qpix robot that was adapted to being what became known as Barracuda. And that became Coastal Genomics, spun off in 2013. And their original, to this day, technology is adapting that actually not into our custom robot, but onto a Hamilton Nimbus again, with custom pedestals and cameras and so forth. And then they built a standalone box called the Light Bench. And then in 2020, the company was acquired by Urgene. So now you can go to Urgene and you can get one of these pieces of equipment. We're looking currently at the Light Bench related to long-read sequencing for size selection there, which is a thing. So they have worked with us, and sort of we gotten very good at this. sort of understanding what you can do with nimbuses, and I'll give you another crazy example. Generally, once you've solved all your problems in the lab with library construction, you need to go off and solve other problems in the hospital, like why is the tumor content in my sample often lower than I would like? Because I dissect the patient, biopsy of some sort, now I have a tumor normal mixture, I want to get more tumor. There's a couple ways of doing this. For long story, but in BC and other public health jurisdictions, sometimes it makes sense to take needles and core the sample. So you literally find where the tumor is, say the tumor is the purple part, as determined by pathologists. You see the pathologist has done some very accurate, okay, to be fair, this is all changing to digital, but it could have changed to digital a long time ago, but it's literally like I was in the hospital last week and they've got their first scanners for actual clinical scanning. Very exciting. Anyway, so the tumor is the purple part. I'm gonna stab it with my needle. I'm gonna inject it into a tube and I'm gonna extract the DNA, and that's gonna be, it's like mining, right? If there's gold on a spot on the mountain, you're gonna dig a hole where the gold is, and you're gonna hope there's more gold under the hole, but you'll see. So we actually, this is such a big thing, thousands of samples a year, that we built a coring robot. And so this is, again, a Hamilton Nimbus, and using their core pipetter head with the expanding O-rings, we have a gripper. And the gripper can pick up these needles. You'll see there in that movie, it's picking up a needle. We've imaged the slide and the block. We've aligned them by the operator doing this semi-manually. And then using these exchangeable pipettes, let's do this again, exchangeable needles, we're actually picking pores individually out of blocks. We can put 24 blocks on this custom deck. This whole robot, by the way, you take that custom deck off in one piece, and it goes back to being a regular pipetting robot. And in fact, due to complex healthcare administrative reasons, we haven't actually deployed this clinically yet. I'll tell you about that some other time. But it's sitting there and currently happily being also mostly working as a regular pipetting robot. So these robots can be pretty versatile if you're gonna be clever. Okay, then COVID happened. Everybody here probably has, we all work in laboratory automation. There's a lot of COVID stories for people who work in laboratory automation. Ours was immediately get asked by the BC Centre for Disease Control for help in the problem of extracting the viruses. Potentially, if they lose their Magmax-based reagent supply for clinical, they need some backup. We were gonna use research reagents, so we quickly developed up an extraction protocol based on our robotics we use for human tissue extraction, and we discovered we had, because we're using now their checkerboard qPCR, highly sensitive assay, way more sensitive than what you do in genomics, we discover we have cross-contamination a little bit. Well, those red things are bad. So what do? Well, we do all the things, because literally everybody at the GSC and the leadership of BC CDC virology is looking at this thing all at the same time. So big ones, really, these are kind of trivial, but change to deeper well blocks. There's a bunch of optimizing pipetting techniques because you realize you have some droplets there, and then actually realizing that your robot moves very quickly, and so you're kind of flinging droplets. So let's not do that by slowing down the robot and getting rid of the droplets. And then actually the custom programming, doing a lot of go down, like for these bead cleans, alcohol, aspirate, bring the alcohol in, dispense, mix, incubate, and then pull out again, and not actually ever come back up to traveling height because you've written a method where you do all those things in one shot. So that was another way of basically reducing the overall robot movement. And then Nimbus specifically has a vibratory tip shoot, and it turns out you potentially, if you have bead carryover in your pipette tips, which is bad, then people were telling me that I was seeing little brown bead droplets in the inside of the robot, which means stuff's getting flung. Bad. So what you do is you actually obtain permission from the very most senior people in virology in the middle of a pandemic to cut holes in the furniture, which was actually, it's a long story, but it was pretty funny. And then I'm very pleased, I personally designed this stainless steel hopper duct thing to dispose of the tips through the table. But really, it seems like getting permission to cut holes in the table, even during a pandemic in a lab, is a thing you have to be brave about. This led to another thing, which was going to those deeper well plates led to this problem of magnet positioning, and this matter of, well, we needed to use this spacer for these deep well blocks to pull the things for the resuspension, and so we'd been playing around with 3D printed spacers, and my guy Dwayne there, he's like, could we automate that? And then I was thinking, you know how you pull a fridge magnet just off the fridge, and what does it do? It falls on the floor, like millimeters away. That leads you to ask the question, how far away should the magnet be from the wells before nothing happens? And the answer is about 18 millimeters. I did this by putting little plastic spacers and testing it myself. And so we built the Maglevitator. This is Angus Wong actually doing the mechanics. This is a Ceres linkage, which is 3 linkages, and one of them is 90 degrees. It's got a little linear actuator, wrote some firmware, a little control box. And so this thing is 50 millimeters tall, so you can use any block on it. It works on the Nimbus, it would work on an Agilent Bravo, for example. Why would you do this? Well, small robots have this thing where in order to use the gripper to put the thing on and off the magnet to make the magnet work, they have to get rid of their tips because the gripper and the tips are on the same head. So you're either being sketchy about reusing tips and maybe cross-contamination, or you're just using a lot of tips. And in fact, in our case, we were using a lot of tips. And so when we did this, we realized we could save sort of like 7 tips per bead clean, but there's, some of our PCR-free has like 5 or 6 bead cleans. Bam. But we rolled out August 1st, 2022 for our main production. We actually have been doing the extractions, which also use beads earlier than that. Anyway, for our AGBT conference in February, I made a poster, so I did the math. We discovered we had saved up to February this year 450,000 pipette tips based on like 20,000 libraries, human genome libraries. So I think, yeah, we're using a lot of pipette tips, so this is not the best either. By the way, I don't like to imagine that whatever I'm doing is like the best thing, and that's why it's really important to come to this kind of organization event and talk to everybody, because lots of people, it's like recipes on the bag that I bought from the supermarket. It might actually work better than the fancy one you got from the fancy cookbook. So, I'm really excited about the stuff we do, but also extremely excited to learn what other people are doing because, I may not have solved the problem the best way. Following the QC theme, by the way, or a little bit, we have, we QC this robot. Angus's stuff tends to work. So we haven't had, touch wood, any failures yet with this thing after getting up to 2 years of operation. But we do QC them every month and they pass. Fortunately, that's led us to other things. Like we have little problems with the Nimbus with potential head tilt. There's some adjustability there. If you lose your parallelism, this could be a problem. So we just built a QC device where we can run a program that basically measures the absolute height between the deck and the head and checks whether it's at all out of parallel. So this is the kind of extra levels of QC that we like to do because, of course, the goal of laboratory quality systems, and we have a whole quality system department with my lovely colleague Maruna, the goal is to not have any incidents and not really have to give them any work because, that's what we are trying to do. Okay, so back to dissection, because this is my latest thing that I'm actually most currently excited about. So again, you have tumor and you have normal. So yes, you can core. Most jurisdictions don't core. They send someplace, a genomics lab, 5 slides with some annotation. Say, like I say, I'm gonna get all the purple parts. Now I've got 5 slides that all look like the thing on the upper right there, or upper left rather. And then somebody comes along with an X-Acto knife and scrapes all that stuff into a tube and then preps it. So you can imagine doing exotic things like laser dissection. The problem with laser dissection is that it's slow. And in fact, you should read my 2020 paper in Journal of Pathology on how slow. It's about 45 minutes per slide if you have a machine learning map of all of the tumor and all of the normal and you've got lots of little patches of tumor tissue, et cetera. It takes a long time, and so it's really impractical. However, we discovered that this is actually an old technique called laser-induced forward transfer, where if you have a coating that a laser can energize, you can actually kind of cause an explosion under the tissue and you could blow the tissue off somewhere else. And so we said to ourselves, and this was our innovation, this is another one of my former engineers, Colin Schlosser, who's now at Accelera, that hey, we could use a scanning laser equipment that you use for engraving. If you ever look at medical devices, they have like stainless steel with words engraved on them, like do not use in humans, like that sort of engraving. Actually, they don't say that typically. So we did that, and we built this thing. And this is a commercial laser engraver and a slide, and with some tissue on it there. And we've just published a paper in Journal of Clinical Pathology, which is actually not as fancy as Journal of Pathology, where we have shown that we can actually do that dissection 100 times faster than with traditional laser capture microdissection. And so we are now looking for kind of like applications for it. And we just managed to get an award from the BC Health Ministry Innovation Fund. You'd be surprised, actually you wouldn't be, how many people in British Columbia Health Ministry have the word innovation in their job title. It's definitely a lot of innovation. So they managed to give me some money for innovation. And so we're gonna do a clinical proof of concept with our collaborators at the Cancer Genetics Lab at the BC Cancer Hospital. We do the sequencing for them, so we're gonna perturb slides. They're the people who love to do coring all the time. So we're gonna perturb some stuff into there. And yeah, it's great. So we're excited about that. There's also, this has implications for single cell genomics, which I'm not talking about today, but we're doing single cells in these custom nanowell plates, or not custom, they're Takara smart chips, little aluminum plates with 72 by 72 array of holes. We're currently using piezo spotting to put cells in wells, but it's certainly possible to transfer material using this laser technique. And so finding, again, use cases and applications for that is kind of an exciting area, but it's a bit early yet. So mostly just winding myself up to, hustle for more money for it. And yeah, so all of this is, in service of genomics, and genomics has really, we've gotten to sort of the point where the sequencing cost barely matters. I mean, a $100 or $200 human genome, I was complaining earlier about the $1,000 medical genetics consult, or, consenting a patient is like this vastly expensive enterprise. So at some point, your little, sequencing cost, or even your $70 library construction cost is not actually that bad. There's also obviously lots of other costs involved in analysis and everything. So really that's a relatively solved problem we have there. We are seeing tremendous growth in our clinical sequencing load because of demographics and because there's more indications for clinical sequencing, and the amount of clinical sequencing is growing in terms of the amount of the genome you want to look at. We're just replacing our 80 gene panel with a TSO 500 panel, but then also everything is sort of heading to whole genome sequencing, and we're getting close to offering a clinical grade whole genome sequencing operation. So that's all a solved problem, and of course, or not solved, it's a solving itself problem. So at the same time, also very excited about long reads, Oxford Nanopore and PacBio. They both have kind of advantages and disadvantages, and they are also providing us with novel and interesting challenges in library construction, things like the balancing of shearing, hydrostatic shearing, and size selection, and then being able to size the things properly. The automation for those library construction methods is actually fairly easy. If you're good at Illumina, you can do PacBio and Oxford Nanopore automation quite well. But certainly thinking about where size selection fits in and shearing fits in. For example, with humans, you want to shear a little bit, maybe, to not have really, really long reads. You want N50s of like 50 kilobases, not messing around with 100 kilobase reads, which clog your pores. But yeah, if you over shear, then you sort of lose some of the value of the sequencing. Anyway, so there's exciting things going on there, but happy to talk more about sequencing if people have projects they're interested in. Thank you very much.
Thanks a lot, Robin. Any questions from the audience? Give you a second to ponder. I guess I'll start off just a little bit. Your tissue dissection thing, I mean, that's brilliant.
Maybe.
Your ability to do that. I mean, any other applications other than the ones that you mentioned? I mean, I can see it being a lot of outside medical potential uses.
Yeah, I mean, definitely. So traditional laser capture instruments are essentially, I think that the use cases early on are like taking almost individual cells. I always think of like Reed-Stenberg cells, and I think it's lymphoma. I'm an engineer, I don't know any of this for real, but they're like bad kids in middle school who try to get other kids to steal gum from gas stations, but in a cancer context. And so they've been studied by dissecting them away from the other cells, or maybe you also dissect that microenvironment around them and try and understand genomically like what people always say they're recruiting other cells to do bad things. So what's happening across that? So these are sort of spatial genomics things, but all of this with traditional laser captures involved like really down at the single cell level. So dissecting across an entire tissue that might be 15 millimeters square for a really large resection is not something that people have traditionally thought about, but you need to recover that sort of thing for doing routine clinical cancer type sequencing. So are there non-clinical applications for dissecting a lot? Well, you certainly probably want to then go image the entire slide and then go do machine learning and figure out like what you want to put where. And so there's some work to be done there. But yes, in principle, there's potentially some research. I know like some labs like OICR does a lot of routine, like Trevor Pugh's a friend of ours, the director of the genomics part there. And I think he'd been excited about the possibilities for this. So it is definitely a thing we need to, we're just sort of figuring out like, we have a kind of version now that we could almost sort of be a beta version for other labs to try.
Okay, cool. Anybody? Somebody's got to have one. There we go.
Hi, just a follow-up question on that one, actually. So when you were removing by laser, you said it was about 40 minutes each sample, but so when you were doing the opposite, does that, like how long does that save in terms of sample preparation?
You mean if you were to use a conventional laser capture instrument, so what we did in the Journal of Pathology paper is we scanned the entire slide, used machine learning to figure out where the tumor was, made a map, exported the map to a Leica LCM, and then we did like 5 slides that are all basically similar because they're all sectioned off one on top of the other. And then that took like 35 minutes a slide, 40 minutes, depending on the thing, took 40 minutes a slide. So now currently that's not being done at all clinically. They would just take a knife and scrape it, or take half of it that looked more tumor-ish and scrape it. So how long does, that takes like a few minutes. Same with coring, right? It takes sort of a few minutes per sample. But then you don't really have like a super precise, you don't have as precise a tumor sample as you'd have. Now, so we're obviously not going to introduce a very long extra step here. But now if I can dissect that thing, first of all, I've scanned, now I'm going to digital pathology. The pathology lab has a slide scanner. So now my pathologist may have actually annotated digitally where some of the tumor is. If you could then use machine learning to figure out where the rest of the tumor is based on that annotation, kind of a directed machine learning. So the pathologist is still kind of running the show there. And then export that map to this scanning laser dissection thing, then I can do 5 slides in like 3 minutes.
Okay, yeah.
Okay, so then it becomes potentially worthwhile. That's actually the goal of this funding from the Ministry of Health, is to can we actually do that? Now the reason, interestingly, the problem is this requires currently a coded slide. So you have to have a slide. So there's some different coatings like metal oxide coating or some polymers that allow you to do this. This has all been known for a long time. And so yeah, we have this problem then where this only works where the lab making the sections can put the section onto the correct slide, actually pointing in the correct direction upwards. In the US setting where everything is separate between genomics and like the block makers, then they have this problem where how do we do this where we can't tell them to use a special slide. So that's kind of the next frontier of this thing, is like what other methods could we use to, sort of enrich or whatever. But in the research setting, also, to some extent you could imagine this thing is like a spatial genomics thing, right? Like I could fire bits of tissue into my nanowells and I can do some high quality sequencing, either because I've got tissue that's spatially interesting, or I just have a whole bunch of cells sitting on a slide, maybe amongst a bunch of debris, and I want to get rid of the debris. So we're sort of figuring out how to push on those in those areas as well.
Okay, great. Any last short questions? No? Okay, thank you very much, Robin.
Thank you. Excellent.
And Robin's going to be around for obviously the coffee time. So please challenge him with some other questions there. A wealth of information there about how to do a lot of different things in the lab. So I'd like to welcome our next speaker is Dr. Sadegh Davoudi. Sadegh got his PhD at the Gilbert Lab at U of T, where he investigated and optimized the use of bioactive hydrogels as an injectable cell delivery vehicle for in vivo muscle stem cell transplantations to increase engraftment capacity. That was followed by a postdoc research into the scale-up automation and further optimization of the MNR assay to create a higher throughput and more accurate model of muscle regeneration. So now as the lead scientist at CCRM, where he specializes in tissue engineering, he is here to share his work on automation in that area. Sadegh, please.
Thank you for that kind introduction, and thank you for having me here. I apologize, I'm recovering from a cold I got last week from my daughter from daycare, and so today I'm going to give you a little bit of a commercial view from where we use automation in specifically process development at CCRM. My talk today, I'm going to start off by giving a little intro about where I work at, and then a quick review of process development and cell and gene therapies, and then we'll kind of showcase the automation platform that we have at CCRM and some examples of what we do there. Now, CCRM is a Canadian public-private partnership that supports the commercialization of cell and gene therapies with strategic funding, dedicated infrastructure, and specialized business and scientific expertise. Back in 2022, CCRM launched Omnia Bio, a subsidiary, which is a CDMO specializing in cell and gene therapies, and it provides industry-leading process and analytical development, manufacturing capabilities from preclinical development all the way to clinical and commercial supply. Now, we do all this with an industry-leading talent pool of more than 300 people with expertise in iPSCs, immunotherapies, and viral vectors. Since our inception, collaboration has been very valued, emphasized, where not only do we work with academia and industry, like University of Toronto and Cytiva, but we also see our clients as collaborators, not just customers. Our collaborative nature exposes us and provides cutting-edge technologies in the space, either through our partnerships or the clients bring them themselves. And of course, to leverage all of this together, we have the specialized infrastructure to do so. Now at CCRM, can you see my pointer? Like that? Yeah, okay. Now at CCRM, we currently have the capability to support preclinical and clinical development. However, as I said, we are expanding. Omnia Bio completes CCRM's long-term vision to have a full spectrum of manufacturing capabilities for the cell and gene therapy space. Omnia will have 2 buildings over at McMaster Innovation Park near Hamilton. One of the buildings is currently complete and operational. The other one is going to be completed in 2027. And together, they're going to add 47 additional clean rooms to the 8 that CCRM currently has. And additionally, the proximity of CCRM and Omnia allows them to support each other, as we've seen over the past 2 years. Now, we've got 3 key areas of expertise, like I said: iPSC, reprogramming and gene editing, immunotherapy development and manufacturing. We work with T cells, NKs, TILs, APCs, and viral vector process development and manufacturing. Now, to support all of this, we have a really strong emphasis on analytical development. We work with our clients to formulate and develop their cryopreservation and formulation strategies. And over the past 2 years, have introduced automation to help with high throughput process analytical development. And also, we offer high throughput media development as well. Now, all of these capabilities transition cleanly into our manufacturing GMP facility, where we have a highly experienced MSAT and operations team. Okay, now that we've got the intro out of the way, I want to quickly go over the role of process development in cell and gene therapies. I'm sure, as most of you know, cell and gene therapies are treatments that focus on using either cellular or genetic material to treat and cure diseases. And to take a cell and gene therapy from an idea to commercial product is a multi-step and complex process. It usually starts with R&D, where you first identify your therapeutic target. And then after that, you take it to process development, where scientists work to create scalable methods to manufacture the therapy while ensuring that it meets regulatory standards, as well as that the product can be produced reliably. And then after that, if the preclinical and clinical trials go well, you're ready for the therapy to be manufactured at scale and being offered to clients, which are the patients in this case. Now, the CCRM's automation platform resides in the process development lab at CCRM. And as I mentioned before, the role of process development is to help move a therapy to the next stage by optimizing the different workflows that the clients bring to us. Now, being a CDMO, we're constantly bringing new clients that have their own processes and ways of doing things. And so our job is to help them optimize their steps. So for example, optimizing their cell culture protocols. Now, that can be a simple workflow like simply expanding a cell line, or it could be a more complicated process of isolating T cells from a leukopak, genetically engineering, modifying them to become CAR Ts, and then expanding them. Or scaling up their process. They want to scale up their process from a laboratory scale to a large scale that's suitable for clinical trials and offering to patients. Now, I really want to emphasize the dynamic and variable environment that we work at in a CDMO. Clients are coming in all the time, and we rarely repeat the same experiment without modifying it for multiple times. So when you want to introduce automation to PD lab, you really need to think about the benefits and costs of doing so. I'm sure most of us love and know very well the benefits of automation. Probably the most important ones is you get consistency and reproducibility. The robot runs things the same way every time. You eliminate user-to-user variability and make sure things work great every time. And probably the most important thing is the throughput that you get with this. Robots don't make mistakes, they work faster, and so you increase your efficiency and throughput. But at the same time, we need to think about the challenges in there. Again, the list is very long, and I really want to focus on the top 3. Biological and viral samples are inherently complex and variable. And so any automated procedure that we introduce needs to be adaptable to different workflows that will need it. Additionally, the next 2 kind of go hand in hand, where adapting existing manual workflows to automation-friendly and compatible workflows needs a deep understanding of the automation platforms and the biology at the same time. And that kind of becomes challenging and costly in some cases, especially because clients that come to us at this point usually have time limitations. They really want to move on to the next step, which is the preclinical trials, and they don't have time for us to spend like 4 weeks, 2 months to create automation procedures. So they want things to move fast, and we're always under pressure to do so. Okay, so with that background, back in 2022, the automation team was tasked with increasing the application of the automation platform in our existing workflows in the process development lab at CCRM. The project was codenamed Wally, and at CCRM, we have the Hamilton Vantage. We also have a Hamilton Nimbus, but I'm going to focus on our Vantage robot, which, as you can see here, is housed in a level 2 biosafety cabinet. Now, on the deck, we have a variety of different accessories. We have an entry-exit module, tilt module, heater shaker, a media fill station, 2 coolers, a variety of pipetting channels, and a 96 multi-probe head. On the back, we have a track gripper, which enables us to move our different labware not only on the deck itself, but also to our off-deck positions, which includes a Lyconic automated incubator, a Citation Plate 5 reader, and a Lab Elite decapper. Now, when automating workflows in the lab, there's a commonly used approach that we use, and I'll demonstrate with an example. So let's say we have this relatively simple workflow, and if you want to do this manually, you kind of do it on autopilot. You don't really think about it. You create your cell suspension from your flask, seed your cells into a 96-well plate, put it in the incubator, and then bring it out when you want to image it on a microscope. Now, when you want to automate it, the first thing we do is break it down into individual steps, or what we like to call tasks. Then the next thing you want to do is go over your tasks and figure out the ones that you want to and can automate. So in this case, we can automate all the steps except the first one, which is preparing the cell suspension. Then you want to start creating your methods on the Vantage and test them and validate them to make sure that they work properly. And after that, you can start combining these different tasks together to create complete or partial workflows. And then again, you want to test it and make sure that it works fine and does what you want it to do. And so at CCRM, we did a deep dive into the different workflows that we have or wanted to have, and identified 8 individual tasks that we wanted to automate. So we have a liquid transfer or addition, liquid exchange, cell seeding resuspension, plate reformatting, which is reorganizing the contents of a plate, serial dilutions or media formulations, imaging, incubations, and aliquoting cell banking. And we can look at these individual tasks as modules, where you can combine these different modules together and create the workflows that we need. And this is a sample of some of the workflows that we frequently do in CCRM. And we can see that we can use these modules to create them. Now, again, this is especially important in the context of CDMO, where time is an important variable, and which directly relates to costs to our customers. And so by doing this, what we can do is rapidly and efficiently create new workflows together. And although there will be some degrees of customization every now and then, but the core of the programming of the modules doesn't change too much. So with that intro, I'm going to give you 2 examples of some of the modules that we created. You can't have a process development lab without seeding some cells and working with cells. So very early on, we knew that we needed to create a module that you could use to seed cells with. So over very lengthy experiments and multiple different testing parameters, we identified the optimal seeding settings to seed different cell types from different reservoir volumes. So you can use either 60 ml or 250 ml reservoir volumes with different starting volumes. And we basically identified the optimal parameters to make sure that you get a homogeneous cell seeding. That includes the mixing height, the mixing volume, the mixing cycles, where to aspirate from, and basically put together this workflow. Now, going back to the challenges of automating a seemingly simple workflow, again, you can imagine the clients that come to us have worked with different cell types, have different requests of how to seed cells and when to seed them, how much to seed. And so we needed to make sure that the method is adaptable enough that can cover most of their requirements. And so we did that. And at the same time, it needed to be simple enough for non-SME users of the Vantage, which would be our colleagues, to be able to use this relatively easily. And so we basically incorporate all our learnings into the method that we created, and at the beginning would ask a couple of questions from the user, like what are you seeding from, what volumes are working with, and then would adaptively change the different parameters throughout the whole process to ensure that you get homogeneous cell seeding from the first well you seed to the last one that you seed. And this is an example of the results. So whether, for example, you're seeding from 25 milliliters or 60 milliliters, we not only ensure that we're getting accurate seeding compared to manual seeding, but also we're actually getting more precision. And so that goes back to the benefit of using automation to improve your reproducibility and consistency. But I think, again, where automation shines the most is your increased efficiency and throughput, where, for example, you can see as you start to increase the number of plates that you're seeding, the amount of time savings you get is more. And then at the same time, and that directly translates to savings for your client, which they care about a lot. Now, the next module that I want to talk about is media formulation. A lot of the projects that come to us, they're optimizing their cell culture workflow. So part of that includes screening for different media, formulating media, for example. And so again, very early on, we developed a customizable module, media formulation module, that would take input file, which contains recipes, liquid classes, volumes, and other information, process it, generate the pipetting patterns, and generate logs for traceability, and then send that information over to the Vantage to formulate the media. Again, the challenges here are that different customers ask different things. And so the method needs to be able to accommodate the variability that comes with that. You have different number of components, different levels for each component. You have different mixing patterns and vessels that the media needs to go into. And so we needed to make sure that the module covers all of that, and that's exactly what we did. So the current iteration of our media formulation module can make up to 192 formulations per iteration. It can make that from 16 components, where each component can have up to 5 levels. And in addition to the reproducibility and consistency, this is again where efficiency and throughput really starts to stand out. So in this case, we were making 128 media formulations from 15 components at 5 levels each, and it takes the Vantage only 4.5 hours to do so. Whereas if a scientist was to do this manually, it would have taken them at least 30 hours, and that doesn't take into account the unknown and known mistakes they would have made along the way. And you can imagine when people come to us, the major cost they have is labor. And so by cutting down this labor, we're reducing costs for our clients, and at the same time, freeing up our scientists for further productivity, because they can do other things in the meantime while they just monitor the Vantage while it's doing its thing. Okay, so those are 2 examples of the modules, and now I'm going to show you 2 case studies of where we combine these different modules together to create workflows. We just talked about media formulation, so let's talk about a media screening assay. Now, in this case, our client was interested in formulating serum-free T-cell culture media that would have similar cell expansion performance as commercially available serum-containing media. This is especially important when you want to take your cells to clinical trials, where human serum and basically serum is not a good thing to have there. And so we needed to create a media screening assay, and we broke it down into 2 parts. The first part would be the media formulation, which we just talked about earlier. Here, in each iteration of the screen, we would make 128 formulations from 15 components, 5 levels each. And then the second part of the screen was a cell culture workflow. It's an automated cell culture workflow, specifically for a 5-day culture of T cells. Now, I want to highlight that every arrow that you're seeing here is basically done on the Vantage. The only thing that is done manually is anything that's outlined in red. And again, this was made by combining the different modules that we created before. So I'll walk you through what we did here exactly. After you formulate the media, we would bring our T cells, thaw them into plating media, load the resuspended cells into the Vantage. The Vantage would plate the cells into a 96-well plate, at which point it would hand the plate back to us to centrifuge it. And then after that, we brought it back to the Vantage. It would remove the supernatant, resuspend the cells in the media that we had formulated previously, and then place the plate back into the incubator. After 4 days, the Vantage would bring out the plate from the incubator, top it up with more media, and then place it back into the incubator. And then the next day, it would bring the cells back out again, add our live dead stain to the cells, and hand the plate back to us, where we would run it on a cytoflex, a flow cytometer, to basically assess cell expansion. Again, everything that you see here was done by building modules, and of the 8 modules that we discussed earlier, we're using 5 of them or variations of them throughout this entire workflow. Going back to the challenges of automating workflows, in this case, for example, we had to take what was done manually by the clients, adapt it to make it automation friendly. An example of that is that if you were to do this workflow manually, at the end of your run, when you wanted to count your samples, you would have a limited number of samples and you could count them with a cell counter on an NC200. But when you have 400 samples in each run, that's not really feasible. And so we needed to come up with a flow cytometry-based cell counting protocol and verify it and make sure that it works properly and that it could count our cells precisely and accurately in the ranges that we were interested in. And that's what we did here. And so I can't do a deep dive into the results as much as I'd like to, but essentially we ran 4 iterations of the screen that we just talked about, where we did 128 formulations per iteration, 4 times. That's we screened 512 different formulations, and of those, 12 of them performed better or at similar rates as our positive controls. Now, our primary metric for performance was cell expansion, but because of the high throughput nature of the work that we did here, we were able to look at other parameters that were involved as well. So cost savings, for example. Of the top 12 performing media, formulation X, for example, was made up of all 15 components, and the cost of that came to roughly $2 per milliliter. Whereas formulation Y, which had similar performance in terms of cell expansion, only used 8 components and came up to 30 cents per milliliter. So the high throughput nature helps us pull out other metrics that you weren't primarily concerned about at the start of your run. And then finally, going back again to efficiency and throughput, we again significantly improve our efficiency and throughput when you automate things if you can do it properly. Now, the last workflow that I want to talk about is an LNP transfection assay. Now, LNPs stands for lipid nanoparticles. They're basically nanoparticles that are composed of lipids that are primarily used to deliver nucleic acids to cells, like RNA and DNA. And probably the most famous applications of LNPs are our Pfizer and Moderna mRNA vaccines. Now, in this project, the client is a manufacturer of LNPs, and they wanted to run a high-throughput LNP transfection DOE assay to find LNP formulations that are able to deliver their payload more efficiently. So we basically had 1,400 LNP samples to test where the payload was GFP mRNA. And again, we worked with the client to modify their workflow to make sure it's automation friendly and compatible with minimal manual manipulation, and this is what it looked like. Again, everything you see here will be done on the Vantage without human intervention. So on the first day, you would basically load your activated and resuspended T cells into the Vantage. The Vantage would seed them into a 96-well plate. Then you would load your LNP library, which are in automation-compatible tubes, into the Vantage. The Vantage would decap them with a decapper, perform a serial dilution on the LNPs, add the serially diluted LNPs to our cells, and then transfer the cells to the incubator. The next day, we had a 24-hour incubation period. It would bring the plate out, add our live dead stain to it, and then hand the plate back to us for assessment on a flow cytometer. And in this case, viability and GFP expression were the metrics that we cared about. And going back to the modules that we created, this workflow was basically put together with 7 of the 8 modules that we initially discussed. Now, what do the results look like? I'm just showing you a snapshot of some of our earlier tech transfer runs where we're comparing manual and the Vantage before we did the whole screen on the Vantage. And so these are 3 sample LNPs. The different colors that you see here correspond to the different concentrations of the LNPs that we created with the serial dilutions. And so starting off with viability, we can clearly see that the Vantage is doing much better than when you do this manually. And so we're already a step forward by automating this whole procedure. And then when you look at the GFP expression in the cells, which basically is a metric for our payload delivery, we can see that essentially they're virtually identical performance between the Vantage and manually. So we're able to automate this entire workflow and actually improve it a little bit. And then, but again, you start to see differences when you look at throughput. As the number of samples that you want to process grows, the amount of time it takes to either create the serial dilutions or create your transfections changes. And again, this leads to cost savings. But additionally, it also makes sure that whether it's your first sample that you're processing or the last one, they're not too different in terms of how long it took you to prepare and work with them. So actually improving your consistency and reproducibility as well. So to summarize, basically when approaching automation in a really dynamic environment, it's really important that we break things down, at least in our experience, into tiny building blocks or modules, and then use those to put together your entire workflows. This enables us to put together workflows rapidly and efficiently, and that's exactly what we did at CCRM. We developed the necessary modules that our clients and ourselves needed to create these workflows and have a proven track record of working with our clients and to adapt their workflows to automation and create reproducible and accurate automated workflows. And additionally, in response to the growing need of automation in cell and gene therapies, we have an internal training program where we're building up on our expertise and creating new and more SMEs at CCRM. Now with that, I'd like to thank, first of all, you guys for coming to this talk, and then my colleagues in the automation team at CCRM, specifically Vlada Sadegh, who was an equal co-contributor to all the work that you've seen here. She's currently in Vancouver presenting this stuff at in ISCT. Our managers, Nish and Paula, other people at CCRM that have helped throughout the different phases of this project, and a special shout out to the folks at Hamilton Robotics, specifically Yarko, Chris, LJ, and Michael, which have really supported us over the past 2 years. I think 2 of them are here today, so definitely reach out to them and talk to them, and I'm sure they can help you out with different things. With that, I'd like to take any questions you might have.
Any questions? 1 second, Robin.
Thank you so much for a great talk. I have a question about the timing. I'm wondering how long does it take to establish a new module, and if you go from one product or one client to another, how much time it takes to...
Yeah, that's a good question. And that's a question that actually comes back to why we created these modules in the first place. So the first time always takes the longest, right? So for example, this was, I think, one of the first media formulation screens that we did. And so it took us roughly 2 weeks to create the media formulation and the cell culture workflow. But then the next client that came along, it only took us, I think, 2 days to change the original and make it compatible for their next workflow. So that's why we really like this modular approach that we've taken here, and try to just make it as a puzzle, so as you fit together the piece that you need based on your needs.
Thank you.
Any others?
I think that was great, thanks. I think you said much more clearly about modules, kind of what I was blabbing about as well. So that's great. I just actually specifically was interested in, do you have any idea why the Vantage was better for viability than the manual? Because it's kind of an interesting result, right?
It was surprising, and I think it really depends on the cell type that you're working in and the environment. So I think we've tested this with serum-containing media. In that case, we didn't see much difference. But in that case, the client was using serum-free media. So the cells are going to behave differently depending what type of media they're in. So maybe, and the Vantage I think is generally gentler than when we do it manually, because I think we've timed, like you set the speeds, right, before your pipetting and aspiration. I think the speeds are generally lower than what you do manually, I think in our experience at least. So perhaps the cells are a little more sensitive when they're in serum-free media, so the lower speeds might be helping, but we don't exactly know, honestly. We haven't, the clients really don't care. So, exactly.
So are you working at all to trying to, decrease that variance, I guess? I mean, you would think that you should be able to get it down. It was what, about 20-ish% or something like that?
I think it was around 10%. Here?
No, sorry, way back on your first one, before the LNP, where you were doing the seeding things and you were comparing it from manual to the automation. I was just wondering if you're looking at making that any better, or if that's just a property of the cell line you're playing with, it's going to be that much variation.
Oh, sorry, in this one. Here, basically.
Yeah, there.
So I think, honestly, well, you can always make things better, right? There's no limit to how much improvement you can make, but I think, again, you want to kind of find a nice balance between how long, like, what is good enough.
Yep, absolutely.
And I think typically we have a plus minus 10% acceptability for error. And so the Vantage matched that, and I think for now that's fine, unless a client comes along and says, no, I really want it to be within 5%. And so then then we'll have to find ways to help them do that.
Yeah, I'm just amazed that the hand one was, the manual one was so good, actually, from that side of things, from things we've seen.
Well,
So,
yeah, Vlada did a lot of the manual testing, and so she's a good handler, so I think she did pretty well.
Great,
I think the average user would be worse than this, I think.
Picking up on some of this conversation, the graph here on the right, I'm wondering if you noticed any improvements or changes in viability depending on the length of the overall assay. I know mammalian cells are sensitive, so presumably faster the better when you're dealing with them being exposed to changing conditions, but I'm wondering if you quantified that at all.
That is usually the case. Again, I think it really depends on the context you're doing the experiments. Serum is really forgiving. Cells really love it, they're comfortable in it. I think you can even leave them for an hour, nothing happens to them. So I think in this case, we wouldn't have seen a major difference, but there are other cases where this time does matter. So for example, if you're doing IT assays, where the time that you add your virus matters a lot. So when you're doing 10 plates manually, that takes forever, and then from your first plate to your last plate, you're gonna start seeing a lot of differences. So it depends on the experiment that you're doing this. Cell seeding probably doesn't matter, it's more of a time savings matter, but then for other experiments that you want to, the time actually starts to make an important difference.
maybe one just last question, anyone? One last one from me. When you're doing your media optimization, have you taken into account using DOE at all for design of experiment
so
for any of it, or is that
I didn't
later
have
on?
time to get into that, but
Okay.
this was actually an AI-based formulation. The client basically had developed an algorithm which it would give you 120 formulations. I think it was DOE-based.
Okay.
It would get the results from us, feed it back into the algorithm, and pump out new formulations which we would test. So over time, the formulations started getting better.
So you're winding up doing like 3 iterations or something?
We did
if you're-
4 in total,
yeah,
and after
cool.
4, we got really good results at the end. Oh, the client got good results.
Right on.
We executed for them. Yeah.
Okay, if there are no other questions, let's thank Sadegh one more time.
Thank you.
And I'll have all the vendors gather over on my left here, just according to your vendor show. And folks, just hang in there. After this, you get coffee and snacks again, so don't go away. This will probably be the shortest time a vendor will ever talk to you. Because I get to monitor and kick them out. Okay, we're going to start it off with Automata. Come on up the stage, please.
Hi, everybody. I'm Bobby Driscoll, part of the field sales team here at Automata. If you haven't heard of Automata, we're a lab automation integrator. What I mean by that is we provide the hardware, the software, and services to tie together a series of automation-friendly instruments into a fully automated work cell. Some of the things that differentiate us from other traditional integrators, stealing from the last talk, we take a Lego building approach to our hardware. So we use small footprint benches that can be configured in a number of different ways to meet the footprint of your lab and the ideal work cell for the automation. From a software, we've built a modern software stack. So it's Python-friendly lab automation scheduling software. We provide developer-friendly approaches. So you can do everything in Python with SDK or API, or we can give you a user experience for like a low-code, no-code workflow building experience. And then third, we're doubling and tripling down on service. So we've heavily invested from the jump in field services and customer success and in our field team to make sure our customers are ultimately successful. So we're over in the corner by the door if you want to come say hello.
Great, thanks a lot. No, I'm just going to, yeah, sure. Next up is Computite. Oh, did I go too much?
Hi, everyone. Nice to meet you all. So grateful to be here. We're a proud sponsor of many LRIG events across the country, United States and Canada. So I would say that CompuType is your resource for anything identifying and tracking within the lab. So imagine that you're in your car and you don't know you could get better gas mileage. You don't know if you need your steering wheel adjusted. But then you have a 4-point system. So think of CompuType as your 4-point system. You need to have your samples identified every time. They have to be able to withstand cryo conditions. They need to be read in downstream automation. So CompuType is here to help you in the lab to get you back to science. So here's our portfolio that I have listed here. We cut our teeth, I would say, on pre-printed barcodes about 50 years ago because data integrity is so important in the lab. We also help folks with preparing labware, barcoding it and marking it so that it's delivered to you all ready to use. And then for folks that want to be able to print and apply on site, we can get you the tools to be able to do that. In addition to the physical labeling that we offer, we also have digital tools such as an inventory management system and RFID. So again, I know that was just a quick introduction to CompuType, but thank you all for your time, and you can visit me in the corner there. Thank you.
All right, thanks. Next up, FragoBio.
Thank you. Hey everybody, nice to meet you. My name's James, this is Landon. I'm coming today from FragoBio, a Canadian distribution company with about 2 million products in our portfolio. It's everything from tubes and tips to high-level capital equipment. We're here today representing and talking about one of our favorite brands, Azenta. This is where Landon's from. We have solutions that fit into many different workflows that you're currently using in your lab and integrate to a lot of the great providers you see around you today. Some of the things we'll have on display at our booth are sample management, both tubes, cryo storage, information about softwares that you can use for tracking your samples through your workflows, sealing, peeling, you name it, we got it. We love to work with you guys closely as customers, but also as vendors with you guys as well. We're here to support you, and looking forward to a great day today. Thank you.
Next up, Thermo.
Well, hi everyone. So my name is Sadegh. So I represent Thermo Lab Automation. So we're a Canadian company based in Burlington, Ontario, with over 30 years experience in automations. So we have a portfolio of robotic movers. So we have the Spinnaker 3 robot, which is a fully collaborative robot, and it's at our booth today. We have the autonomous mobile robot, the Inspire vertical collaborative platform, and we have a broad range of automated cytomat incubators for microplates and storage solutions. And if you're working, if you're doing cell culture in T-Flask, we have the Celtrio cell line automation that can handle T-Flask incubation. And finally, we have our momentum scheduling software, which is very flexible and powerful with over 450 instrument drivers, allows for real-time data connectivity. So we combine with our portfolio with our expertise and knowledge and automation, we can help you accelerate your science. So feel free to stop by our booth today. Thank you.
Great, thanks. ReadySoft.
Hello, everyone. Thank you for coming today. I am Joyce Padilla representing ReadySoft. So we are a automation company, Canadian-based, and we've been supporting lab automations for over 25 years now, helping them to accelerate their research and processes. Sorry, I don't know what happened there. It's all good. So one of our flagship products is Genera. So Genera is a dynamic scheduling software that really is known for its flexibility and reliability. So sometimes when we're talking with our customers, we tend to ask the question, what are you looking to automate? Because we've proven over and over that Genera is very flexible due to its open architecture, that we're really able to accomplish a lot. So if you find those tedious jobs that you don't like doing in the lab, feel free to reach out to us as we can help you automate the process. Another product we have is our integration solution, XLab. So basically from a small integration that you may be starting out into lab automation to our full complex integration system. we can definitely help you support that with our XLab product, which has everything you need to get started, and it's designed to be scalable. So as your automation needs grow, so can your XLab solutions grow as well. And apart from that, we really strive and really care to provide a really good experience to our customers. So we've created a hub, it's called ReadySoft Hub, where we're able to empower our customers to learn and educate themselves on demand about Genera. Thank you so much, and please visit us when you can to get more information. Take care.
Coope.
All right. Thank you, everyone. I'm Kayla Smith with Coope Scientific. I'm very new to the team, only about 2 weeks in. But Coope Scientific is a supplier of certified pre-owned automation equipment, general lab equipment, as well as imaging, so all the way up to confocals and other imagers. We also provide maintenance for all of those instruments as well. So when you think of pre-owned equipment, a lot of people think it's something that's flipped from an auction. One of the things we pride ourselves in is that we actually refurbish the instruments and can customize to whatever you guys are looking to do within your lab. So the name of the game this year has been cost savings across the board. So if you guys want to, make your budget stretch as far as you possibly can, Coope is a great company to work with. In addition to that, any of the certified pre-owned equipment also comes with a 1-year service warranty for automation, which is kind of unheard of for pre-owned. So we are pretty much agnostic to any supplier. We can service Agilent Systems, Beckman, Hamilton, TCAN, and a number of other automation platforms as well. So if you're looking for a certified pre-owned servicer or integrator, feel free to stop by the booth. Thanks.
Thank you. Next up, Hamilton.
Hi, everyone. I'm Michael Saul. I'm the Director of Sales for Hamilton Robotics here in Canada. I'm here with Louis-Jacques Forte, who's my automation consulting engineer. You got any technical questions? He's here to talk to you. Just a little bit about Hamilton. I mean, we offer liquid handlers, sort of the backbone of a lot of the things that are being done, from standard solutions to custom solutions. We do that through our patented technology. We have quite a few technologies that go into the systems that make them unique. We've got great software that's extremely flexible, which I think you've seen with some of the presentations here, people being able to do some really cool things. We have quite a big sort of consumables portfolio as well, so different consumables for different applications depending on what you're doing. And I think really the most important is the continuous research and development. I think if you go to SLAS and you see Hamilton's booth, you see new products every year. We come up with new and crazy things all the time as we're trying to support you. The last thing I wanted to say was, if I do know you, if I've worked with you before, it's been an absolute privilege. And if I don't know you, come by the booth. Maybe we can work together. Cool. Thanks.
Thanks. Hudson.
Is this on? Okay, good. Hello, everyone. My name is Karina O. I am part of Hudson brand of instruments and equipment. I am coming from Boston, Massachusetts. My company actually has 3 sister companies under the name of Hudson. The first is Art Robbins Institute, which has been known throughout for 20 plus years in regards to protein crystallography, but also the liquid handling and ability to go down from nanoliters up to microliters. The main one that I am actually been the most familiar with is Hudson Robotics, which is based out of New Jersey. Hudson Robotics is an engineering company that is basically wants to help you walk away, give you time to do other things, just get rid of the repetitious tasks that you have in regards to a couple of different core competencies. One is robotic handling of microplates, liquid handling, as well as things like the colony picker, And then TomTech, which is a company that we just had acquired. They actually do LCMS sample prep as well as other liquid handling capabilities.
Thank you. Dynamic.
Hi, everyone. So Dynamic Devices has a liquid handling robot named the Lynx. The Lynx has different options. One of them is the 96 VVP head. The 96 VVP has a unique feature where you can have 96 independent volume, meaning the tips can aspirate or dispense different volume all at the same time. So it's perfect for normalizing a plate for a specific volume or concentration. Another option is the MCPA. The MCPA enables the robots to change the tools on the fly. That means on the deck, you can have different heads, let's say a 24, 96, 3D4, and the robot can go and pick up the heads that it needs on the fly. We do offer as well other tools, such as the 24 or 96 magnet rods for beads purifications. So if you want to know more, please stop at our booth. Thank you.
Great, thanks. TCAN.
Good afternoon, everyone. I'm Busy Oliver, and this is David Sullivan. We're from TCAN. We are the first company that came to market with an automated liquid handler in the 1980s. So on the far left there, we also have a lot of reagents, diagnostic assays, but our system is kit agnostic in general. We have a detection line of products as well that can be used as a standalone or integrated into our liquid handler. I'm sorry, I'm not talking into this well, am I? The liquid handler has a lot of different variations from some of the others on the market, so we really hope that you drop by and hear a little bit about what we have today. We are the booth on the far left over there. Thank you so much.
Great, thank you. Festo.
Thank you, Tom. Good afternoon, everyone. Great to see everyone here. So my name is Ross Bilton. I'm with Festo. Festo is an automation company, been in the market, global market for 99 years. We focus in two areas, technical education and automation. In automation, what we focus on, particularly for life sciences, is the core of processes, liquid handling, motion and control, gas handling. The way we go to market, we work with our customers in applications, so it could be anything from total lab automation as examples, analytical instrumentation development and design, or pre and post analytical design. We provide the automation products. So we make the components for gas control, motion control, liquid handling, basically 30,000 different products that we work with our customers how to put them into a particular application to solve automation challenges. So to make reference to Robin and Sadie's comments earlier, if you're into innovation, we like to work with you. And as Sadie presented in his presentation, we consider ourselves the Lego blocks of automation. So when you need to build something, we have the catalog of components and the engineering expertise to help with that. We're right here. Come see Ed and myself. Great.
Great, thanks. Formulatrix.
Hello, everyone. I'm Brandon Weber, representing Formulatrix. Formulatrix, protein crystallography, filtration, but we're here talking about our liquid handlers today. I think in the essence of time, if you're looking for automation needs for your liquid handlers, we're back in the back. Swing on by, and we can kind of further discuss what your options are, wide flexibility of our instruments. So stop by the booth in the back, and we can further discuss if you need any automation needs. Thanks.
Great, thanks. Planar.
Back off. Sorry. Back off. That's okay. Steve Yardley from Beckoff. I'm here with my colleague Paul as well. We're over on the side there. So Beckoff Automation specializes in open automation systems using PC-based controls with products including industrial IPCs, I/O field buses, drive technologies, automation software, and hardware. Today we're highlighting our XTS and Explainer technologies. So Explainer should be of high interest to anyone looking for lab automation, as the solution combines the advantages of your conventional system and supplements it with the unique magnetic levitation technology. So with its floating 2D transportation, the planar motor system opens up a whole new host of possibilities. User benefits from individual freedom of movement, the ability to transport to any location via any route within either a single machine or between several. Easy to deploy and redeploy as you evolve. Explainer simplifies individual production steps considerably, making the degrees of freedom and positioning accuracies familiar from robotics by using 6 degrees of freedom. The transport system itself, it handles the positioning and product during the process. Cleaning and maintenance requirements are drastically reduced as there's no moving parts and it's virtually silent. Come talk to us and see how we can help your lab become more efficient and how to build the lab of the future. Thanks.
Great, thank you, Beckman Coulter.
Hi everyone, I'm Samuel Santerre. I'm the Beckman Lab Automation and Genomic Consultant. I'm supporting the Echo acoustic liquid handler that use sound wave to transfer liquids, the Biomech liquid handler, robotic integration, and also the genomic reagent using Sprite Beat technologies. We recently launched the Biomech Echo One, which is in the top right corner. It's combining best of both worlds, the Echo and the Biomech together, giving a wide dynamic range of liquid transfer from 2.5 nanoliter to 5 milliliter. So if you're curious to know more about the acoustic droplet ejection that use sound wave, how it can miniaturize assay, save expensive reagents, and do direct dilutions that are more precise than the regular serial dilutions, and how this instrument can reduce plastic waste in your lab, please come visit us at the Beckman booth. Thank you.
Great, thanks. Biodot.
Hey, good afternoon, everyone. I'm Michael from Biodot. So we offer low-volume liquid handling solutions for picoliter, nanoliter, and microliter volume ranges. All of our equipment is built at our headquarters in Irvine, California, and our equipment's very versatile and flexible, so it's used in a wide variety of applications, such as genomics, cellomics, proteomics, biosensors, microarrays, just to name a few. And we're part of ATS Group since 2021, so we can now offer lab scale dispensing automation all the way up to high throughput. And we are right over there in the corner. If you have any questions, please come on by. Thank you.
Great, thanks. And Opentron, sorry.
Yeah. Last but not least, going to try to get up here and not sound like a robot, no pun intended. Hello, everybody. My name is Tyler Smith. I'm the automation specialist for Canada here at Opentrons. At Opentrons, we specialize in providing scientists with accessible pipetting robots. That means that our robots are affordable, they carry a low cost of ownership, but also that they're easy to use. This enables small research teams to large research institutions to automate tedious pipetting tasks that would otherwise be done by hand. Customers often comment on the fact that they like that we are reagent agnostic as well as labware agnostic, and the fact that our firmware and software is open sourced. This just enables the robot to accommodate for your science versus you having to adjust your science to the confinements of a traditional liquid handler. One thing that is coming that I wanted to mention, we are leading the forefront of a codeless experience when it comes to pipetting automation through our partnership with OpenAI. Be on the lookout. We have a tool coming out that's pretty similar to ChatGPT, but to create protocols on our robots.
Cool. Very cool. Okay, great. Thanks a lot. Thanks for hanging in there, everybody. Like I said, it was short snaps of what these vendors around here do. We've got coffee, cookies, and there's still some lunch left. Please grab something. Please spend the time, talk to these vendors, and stick around here until our last talk, which will be happening about 3:15 to 3:30, and we'll learn a little bit about how automation is used in chemistry. Great. Have a good mingle. speakers before you.
Hi, my name is Organa. I am a robot chemist. Can I help you with anything today?
Hi, Organa. I would like to run a
Could you please specify what the experiment is? Hi, my name is Organa. I am a robot chemist. Can I help you with anything today?
Hi, Organa. I would like to run a chemistry experiment.
Could you please specify what the experiment is?
I would like to generate a Torbay plot for an unknown quinone solution.
What is the setup of the lab environment? What are all the reagents and vessels?
So in my lab environment, I have a 10 millimolar solution of an unknown quinone. I have water, I have a 1 molar solution of sodium chloride, and I have 0.5 molar solutions of pH buffers ranging from pH 4 to pH 9. I also have 2 beakers, one experiment beaker and one waste beaker, a potentiostat, and a pH probe.
What is the rationale behind the experiment?
So the goal of the experiment is to measure the potential of the quinone solution at various pHs. We'll start at pH 4 and take a measurement there, and then we'll scan from pH
Hello, everybody. We'd like to start with the last presentation, so if everybody could just finish up and grab a seat, that would be great. That includes you two vendors. Come on over and grab a seat, sit down. Wander over. Vendors, if you want to come and grab a seat too for a change of pace, be one of the audience, that'd be great. Okay, it's my privilege to introduce Dr. Alan Aspuru-Guzik. Alan received his PhD in chemistry at UC Berkeley, and then he moved to a professor position at Harvard, where he worked at the intersection of theoretical chemistry with physics, computer science, and applied mathematics. He's now moved to his current position as professor of chemistry and computer science at the University of Toronto and the Vector Institute, where he holds a Canada 150 chair. He has co-founded many companies where he's focusing on research on quantum computing and AI for materials discovery and energy. His work aims to revolutionize chemical sciences through technology innovation. And with that, I'd like to introduce Alain.
Okay. Okay, can you guys hear me here? Okay, so I guess I have 25 minutes, and I have more slides than 25 minutes, so I'll be skipping some slides accordingly, okay? So don't get too dizzy. But today I'll be telling you about a topic that we're very excited about here in Canada, especially at the U of T and UBC. We have a big initiative I'll tell you about. We like to call self-driving laboratories. So maybe to find out the audience, how many people have heard of what is a self-driving lab? Okay, it's about 50%, that's really good. So for the other 50%, let's just begin, right? So about 5 years ago, I wrote this paper with my other colleagues, Marcus Reicher and Roland Lind, thinking about what was the future of theoretical chemistry in the 21st century. So I started as a computational chemist, and when Trump was elected, I decided to move to Canada. I was living in Boston before. And when I moved here, I also wanted to do experiments. Why? You will see, but I started thinking about the fact that computational people and experimental people are too far away from each other, and that all the robots that we see here need a brain, right? They're pretty dumb, sorry to Hamilton or to Opentrons or whatever, pretty dumb things, right? They're just moving things around, but they really don't have any AI on them. They're not really planning experiments, discussing what the output of experiment is, and proposing the next experiment, right? They're just a machine that carries out experiments. So they are kind of like a very nice robot, but it doesn't have any brain, right? And now, how many of you have used ChatGPT or any LLM? Raise your hand. So can you see the connection? Can you imagine what can happen if you have something like an LLM and one of these machines together? More or less, that's a self-driving lab. I'll try to define it much better, but think about real AI connected to these machines. So I wrote this paper where I said, well, I mean, there is this thing called Organa. You'll be able to talk to Organa. Organa will make you molecules, will synthesize and characterize the molecules, okay? So I'm gonna skip this. It's not relevant here. So the dialogue will be like, hey, Organa, suggest to me a molecule. What are the symptoms? I'm very interested on like some of the machines that I see here that are more biologically oriented. I'm interested in making molecules, so I need reactors and pumps and stuff like that. That is a little bit different, but it's the same idea, right? And you could also imagine reactions that are harsher than the reactions that you can do like in these 96 well plates, right? These are reactions that require a little bit more automation. So anyway, this dialogue that I'm skipping goes back and forth and imagines this dialogue between a machine and a human. And it's only 5 years after that, almost 6 maybe, that I published the paper, and now we have the capability of very simply talking to machines, right? So let's talk about that and planning and all the things together. So, well, self-driving lab, like what's the idea? You have chemical space or formulation space or material space or biological space. They are usually huge combinatorial spaces. The number of molecules that you can synthesize is about 10 to the 180, larger than the number of atoms in the visible universe by a factor of 10 to the 100, right? It's almost impossible to even imagine how many molecules you could make. So when people were doing high throughput chemistry in the 1990s, 2000s, they were bound to fail many times because they were exploring chemical space very inefficiently. Basically, what a self-driving lab is a lab that is driven by AI where every experiment counts. But of course, we're in an automation conference, so let's talk about the history of automation. I mean, this is perhaps one of the first chemical automation experiments. The automatic synthesis of peptides gave Bob Merrifield the Nobel Prize, right? So we start with the 1960s. I was born in 1976, so this is 16 years before I was born, right? This is kind of where automation, I think, seems to have started, right? But then it's very interesting. What was the first self-driving laboratory? And I don't know if you guys can see my slides very well. I think my background kind of sucks, but you can see here what we like to have now in many of our papers, this idea of the closed loop optimization or closed loop feedback between an AI and a computer. Well, the earliest version I could find is this paper from 1978. I was 2 years old, which basically the abstracts looks very similar to the abstracts of our journal Digital Discovery, which actually talks about this, right? So now there is a journal in the Royal Society of Chemistry. I am the editor-in-chief, all about this field of self-driving labs. So, but you know, this is basically the same idea. There's an optimizer coupled to a basic programming language, and it's controlling some chemical pumps. That's pretty cool. This Winnicott guy is still alive. Actually, was tracked by a reporter, and he used to work at SK of GSK back in the 70s. So, modern version of a self-driving lab is on the left-hand side of the slide. You will have an optimization algorithm on some sort of AI. You can see there a neural network, for example. On the right-hand side, you have these platforms. Obviously, the biggest challenge is that some of these platforms have very cranky software interfaces, very old APIs, right? So what we like to do is think about the ones that are pretty interactive, right? And some of the vendors here have better access than others, okay? So think about that when you're trying to build a self-driving lab. You have to be able to enter the machine with your software and control it, right? So now, again, high throughput experimentation, 1990s, kind of not so great idea, usually, right? I mean, what's the point of enumerating experiments that you know might fail, right? It's much better to think about what are the experiments that give you the maximum amount of information about your system with the least amount of trials, right? So that's what we do. And actually, Ilya sitting here, actually, you can talk to him also later. He's done these experiments in the biologic setting, in organoids and drug delivery and so on. So you can talk to him about how to use AI in those kind of settings. If you're interested in biology, we have collaborated on that. So if you want to learn more about self-driving labs, besides my talk, there are 2 papers. One that finally we got the reviews back is submitted to ChemRev. It's in the top, it's in the Chem archive. It's about 600 references, so you can download it and see more about this. And the one that I'm really excited about is already published, and it's in Digital Discovery. It's about the frugal laboratories. Part of this talk is called The Revolution Will Not Be Televised, because I'm very interested in self-driving labs that you can build with very inexpensive parts, right? So you want to use like very cheap parts to actually build your own self-driving labs. For example, this is an example of the Jubilee platform, I think, from, how many of you have heard about the Jubilee platform from University of Washington? Really cool, bottom up. They take like a stage for a 3D printer and convert it into a high throughput machine, not very dissimilar to these machines. So you can download the parts and just start building your open hardware. What are the advantages of building your open hardware? You can do anything you want. So my lab just published a digital pipette. So we have a pipette made literally with a syringe that is as good as a digital, as a regular pipette, right? And we built it for $200. So a lot of the stuff that we're doing nowadays has to do with that. But you also see their computer vision. Why would you care about computer vision? We're going to move from coordinate-based systems to computer vision control systems, right? I have some videos I'll try to show you later that are really cool about that. See if I have time. But frugality. The entire startup of my former postdoc, now professor at UNAM in Mexico, was $5,000, right? What can you do with $5,000? You cannot even buy an OpenTrons, right? So what he did is basically build his own system with these pumps with $300, right? So these very simple, cheap pumps, and he was able to do some simple condensation reactions, very useful for teaching. So that review that I have is all about this kind of frugality. Can you make the simplest possible self-driving labs with cell phone cameras and stuff like that? But now, of course, we need an operating system. So how many of you use an operating system to control your automated systems? Some sort of orchestrating system. There's about 2 of you, okay? So I think 98 remaining of you should actually think about that, right? What is the data infrastructure that's going to take the data in and out, right, of your self-driving lab, store all the experimental data, and decide what is the next best experiment? So we released in about 2018 our first version called ChemOS. There are other ones there that you can see here listed here. There's one called oh my God, Benji Maruyama's, I'm forgetting the name of his one, but here are the references. There's 2 more or so, right? That we're trying to integrate, you know, how to handle self-driving laboratories. Obviously, that means computation, data storage, extraction from these machines, talking to the machines, etc. Now, since then, we were awarded the largest grant ever given to any Canadian institution, 200 million Canadian dollars. All basically here at the University of Toronto, which basically matched and exceeded that donation. So this is about a $500 million investment, right? So we're building this building. If you guys walk around U of T campus, you will see a hole. This building is gonna come up. It's basically gonna have 3 stories full of self-driving laboratories. Ilya's self-driving lab is gonna be there, hopefully. And for that, we need a real operating system. We need to control what's happening in labs that range from inorganic chemistry all the way down to organ on a chip. Right? So we need to kind of control all of that data. So that's where ChemOS 2.0 comes out. The paper is already published in the journal Matter. I apologize, I haven't updated these slides. But many of you have heard about the idea of edge computing, like sometimes you run neural networks on your phone, or they are running even in your like temperature controller in your Nest or things like that, all the way down to a supercomputer or the cloud, right? So the analogy here is obviously we have our databases and our software. The fog is going to be some PC or some computer connected to your machine. It could even be a Raspberry Pi. And then the edge might be the actual robotic arm or the actual liquid dispenser or whatever, liquid handler, right? But obviously, you have to also orchestrate other things, like, for example, optimization algorithms, right? Or simulation databases, or also orchestration, right? Like, you really want to know who does what next. And in my labs, and all of the labs here at U of T, we actually, of course, rely a lot in soft robots, which are basically called undergraduates, that carry things between robots. So don't imagine our labs as these places where everything is automated. We believe that you have to automate what needs to be automated, other stuff. You just have an undergraduate carrying plates around, right? But we use an interface called SILA. How many of you know about SILA? It's a interface developed in Europe about controlling this machine. So we use SILA. So we try to be kind of standardized, and we're trying to work with people from Argonne National Labs and now Cambridge or Oxford, sorry. They're developing also other self-driving lab software to try to see if we can unify all of them into a single one. So anyway, that's the story. You can have some sort of simple web interfaces, and the paper has some examples, okay? So just as an example of a bottom-up self-driving lab, let me just tell you how we use ChemOS 2.0 to do electrochemistry. But also we want to do it frugal. How many of you do electrochemistry here? Automated? No? So you probably haven't seen, let me show you. You probably haven't seen biologics machine. You know, this machine's $15,000 bucks. Basically a good electrochemistry setup. If I want to have 64 channels of biologics, I'm already burning out a lot of cash, right? So a lot of the things that we think at U of T is how can we actually engineer around them. So we built this $120 PCB board, okay? That basically is called eToad or Open Potentiostat that actually gives us, of course, crappier data, but way cheaper. And we also connected it to an open synthesis platform that we call Medusa, okay? How does Medusa look like? Medusa is a bunch of pumps, a bunch of reactors, a bunch of electrochemical cells. We have different ranges of electrochemical cells. Here is our open potentiostat. So why am I arguing for you, perhaps in industry is not relevant, or maybe other people in academia might say, why I want to just hack everything? Well, because we're trying to make everything very modular. Once we have a system, for example, we have an automated robotic electrode polisher. That system can be used for many other settings, right? So we're trying to make everything software and hardware modular. How much time do I have left? Probably 5-10 minutes? A few more than that. Okay, sorry. So let me just show you one example really briefly. It's about 8 slides of what we have done already with a self-driving lab. It was just published in Science 2 weeks ago. So I haven't updated these slides with the paper, but you can download it from Science. So the story, because I don't have too much time, is about the biggest fraud, almost, in physical chemistry. How many people have heard of Hendrik John? Right? Oh, Robin knows about him. He was a big fraudster. This book is amazing to have in your toilet when you read about it. It's a great book because it talks about how easy it is to fake data. I mean, the self-driving labs and all this automation hardware allows us to make sure data is reproducible, right? But this guy was faking data at Bell Labs at the highest levels, and he invented, supposedly, organic superconductors, semiconductors, and organic lasers. There was this postdoc that was instrumental in debunking all the bullshit this guy was doing. And his name is Chihaya Adachi. He's a good friend of mine. So he told me about 2017, Alan, if you want to make a self-driving lab, do it with organic lasers. My friend Marty Burke told me, let's do it. So we started doing organic lasers. Very strange thing. So your cell phones have organic LED. You spend about 6 hours a day looking at your OLED, okay? About in average. Can you imagine that? Probably you spend 12 hours, I don't know, right? Like this. Well, if it was a laser, it will use way less power. It will be directional. It could look at your eyes and just shine light at your eyes. Sounds weird, but that's the next-gen tech. But at the time that we started the self-driving lab for this, there were only 10 published or so. So we said, okay, bring it on. So we started thinking of a modular synthetic platform. This is harder reactions than regular reactions for pharma because these are like very hydrophobic molecules. We're using metaboronate protected Suzuki couplings where we changed actually the protecting groups. There's a lot of chemistry that went into this. But then what we did is we integrated a ChemSpeed platform, which now I don't use very much because now I build my own synthesis platforms with with Medusa, right? I can build something that does the same thing as a ChemSpeed for $5,000 bucks, right? So why would I do a ChemSpeed? But at the time, of course, we bought a ChemSpeed. We bought a big fancy HPLC and also a laser optical table for characterization. So the way our self-driving lab looks like is like an organic chemistry, analytical chemistry, and physical chemistry lab controlled by a theoretical chemist, right? It's kind of like a weird thing, but it can do time-resolved spectroscopy and synthesize about 40 molecules, say, in about a couple days or so. Synthesize and characterize. The bottleneck is really not the synthesis, but the characterization. You have to feed these things into HPLC. Takes a long time to separate them, and then do the optical characterization. Takes no time, but it's basically bottlenecked by the fact that we have one HPLC. Okay? It's all fed into the HPLC automatically. To make the story short, I mean, people like to see robots. I really like this integration that we purchased. Now we could build it ourselves, and my friend Jason Hine at UBC builds these things with the eyes closed. But we purchased this thing from Quantus Connect. It's the best powder dispenser I know in the universe. And then we have our ChemSpeed. But you guys have seen those things before. What probably you haven't seen is the Medusa. This is where we did about 95% of our reactions, our home built. You can see the heater there, like these disposable vials. And you know, we do all our reactions there. Turns out it's easier to do them there than in the ChemSpeed, right? So you see basically here our two-step one-pot synthesis happening in this thing. Pretty cool. Now I want to skip, well, we basically, to make the story short, synthesized about 1,000 organic lasers, okay? So this is about a factor of 100 more than everything that was published. Ran it by a Bayesian optimizer, and maybe without, seriously, I'm trying to compress a lot into 25 minutes, but we're trying to optimize, this is a number of materials that were better than the state of the art. Let's just think about it that way, okay? I'm simplifying a lot. The point is, like you can see here, when we were fiddling with the robot, we were discovering very nice molecules. Then we did what is called a hypercube sampling. Have you heard of Latin hypercubes? Basically, you take a data set and you say, okay, what are the most diverse points in the data set? If you guys were a two-dimensional data set, I will throw a point here, here, one there, one there, one in the center, roughly, right? But of course, in a higher dimensional space, I mean, I really space around a 300, you know, it's actually 190,000 molecules, but I have the 500 molecules that are as diverse as possible in that sense to train my Bayesian optimization model. And then after I ran those 500 experiments, then I started running my Bayesian optimizer, which is the core of the self-driving lab, is the one that says, okay, this is the best experiment to make, either to learn something about it, or exploit. And after running about another couple hundred experiments, we had enough materials to actually write a science paper. So we stopped, right? And these are the 3 best organic lasers in the world. And that's how they look. Okay, so how much time do I have left? 7 minutes, perfect. So now I want to spend my last 7 minutes telling you about the future. Well, what is the future? So we talked about Organa, right? So I have a very controversial argument that we can discuss in the Q&A, but if there's Q&A or offline, I feel we should use, instead of trying to fit our chemistry into these automated machines, we want to fit the automated machines to our chemistry. What do I mean by that? Okay, all the organic chemistry literature that we have is done at certain scales, at certain, human-handled, glassware, right? And also in the developing world, the labs already look like that, right? So you don't want to spend $2 million or $1 million outfitting a lab to look like a bunch of liquid handlers, right? Maybe you want to think about differently. You want to think about a humanoid robot. Now they cost $20,000, the cheapest ones, right? Entering your lab and starting to do experiments with the stuff that you have. That is the vision that we're working on, right? So to start that, we have to come up with vision. There's a lot of automation without vision. So we started working about 3 years ago or so in the problem of transparent object detection. It's a very hard problem in computer vision, right? Because glassware is usually transparent, right? So we started building these large data sets that actually use computer vision to actually detect transparent objects. And 2 years ago or so, we had the state-of-the-art computer science work on this. This is my collaborators in computer science really drive this. Florian Scrutin and Animesh Garg. I am the chemist in that collaboration. But we can detect the bounding box of this glassware. We can pick it up. We can see it. We can manipulate it. We can even estimate how much liquid is on it. Extremely useful, right? So this is another, for example, another piece of work, multi-view trans. We actually are able to use many different perspectives of the robot. Sorry, I guess this got, sorry, like this video is like, never mind, I'm gonna skip that. And also optimize trajectories. For example, like this is a very early simulation that we did in our digital twin of our arms not spilling chemicals, right? So you can even think about manipulating chemicals. We started with pouring, sounds weird, but now we have digital pipettes. Now we're working on digital solid dispensing, all of that bottom up, 3D printed, adapted to the robot arms that are cheaper and cheaper and cheaper, right? You can even buy a Chinese version of these robot arms for about $5,000 now. We buy the European version for many reasons, but basically you can see here early versions of our experiments where you actually are doing solubility experiments and so on. What is nice about this robot is that you can see everything is instrumented connected to ChemOS. For example, this balance that has a heater and basically a planning system. Because if you want to think about this kind of more general problem of pick up that flask and take it here, you have to do robotic planning. You have to think about what is the order of events you're going to do it and also the trajectories, right? This is not now X, Y, Z trajectories. This is, I had to pick up something on the real world and take it somewhere else. So this is where LLMs came in, and I'm running out of time, I can see. But where LLMs came in, we started thinking about how many have you heard of hallucinations in large language models? Probably many of you, right? Hallucination in this would be catastrophic, right? You run an LLM and says, okay, mix this with this is explosive or toxic, right? It's not possible. So you have to actually do verification. So we published a paper last year on how we can have this like structured verifiers that whenever we generate this chemical language to actually do these experiments, the verifier actually makes sure that it's not BS, it's actually an experiment that can be done. It doesn't have too much safety in it yet, but it has validity. But the cool thing about it is that, of course, you can imagine that you can talk to ChatGPT and tell it to, make a Midjourney will make a picture of a person and ChatGPT will give you a poem, right? You could also ask it now to actually do an experiment, right? And maybe I will end with that, actually. Let me show you what the state that we are. I told you that 5 years ago I was thinking about Organa. I was thinking about the system that was going to do experiment. So this system is very contrived in the sense that it can only do about 4 classes of experiments. But every module on it is extremely generalizable. Okay? So what I will show you is very cool. It has a planner, and the planner that interacts with LLM is trying to figure out what are the stages of an experiment. What is the setup? What are the things that it needs to do? What is the desired outcome? How many experiments should it do? But because it doesn't know that, it's talking to a human. The human could chat, could give it in text. But in this case, Marta and my students actually made it super cool. They actually made it voice controlled. The only thing that you will see here is 3-second delays that we cut off between when Marta talks to the robot and all the processing happens, because they are annoying to see in the video. But I'm going to end this talk by showing you where we are with Organa, okay? So this is kind of how we think the labs of the future will look more and more. Obviously, this is not going to be a fixed arm. It's going to be an arm that is walking around, right? With 2 arms and robot that's doing things. But at the moment, this is more or less how the vision is.
Hi, my name is Organa. I am a robot chemist. Can I help you with anything today?
Hi, Organa. I would like to run a chemistry experiment.
Could you please specify what the experiment is?
I would like to generate a Torbay plot for an unknown quinone solution.
What is the setup of the lab environment? What are all the reagents and vessels?
So in my lab environment, I have a 10 millimolar solution of an unknown quinone. I have water, I have a 1 molar solution of sodium chloride, and I have 0.5 molar solutions of pH buffers ranging from pH 4 to pH 9. I also have 2 beakers, 1 experiment beaker and 1 waste beaker, a potentiostat, and a pH probe.
What is the rationale behind the experiment?
So the goal of the experiment is to measure the potential.
So you see, she just, at this, speaking as fast as she spoke, narrated all the elements, and the robot is like, okay, pH probe, I know what a pH probe does, I know what this and that, and it's already organizing what things are available to it. So it's already the inventory. Okay, and now it's trying to figure out what the experiment is for.
Of the quinone solution at various pHs. We'll start at pH 4 and take a measurement there, and then we'll scan from pH 4 to pH 9.
Could you please provide an example of how to run this experiment?
Yep. So to run this experiment, I would first add 6 milliliters of pH 4 buffer solution. Then I would add 3 milliliters of sodium chloride solution, followed by 15 milliliters of water. Finally, I will add 6 milliliters of the quinone solution. Then I would measure the pH and run a CV scan. And finally, I would transfer the contents from the beaker to the waste beaker.
Can you tell me what the expected outcome of the experiment is?
So it's getting a little bit tedious, but I can't fast forward on this computer right now because it's connected offline. But maybe I'm thinking, how much time do we have left? Probably not too much. A minute. So let me just then tell you what happens, and maybe I'll try to, well, maybe as I'm talking to you, I'll lower the volume, and I'll tell you some things. Because, yeah. So I want to tell you things at the same time this is going on. So what's going to happen then, that you will like a lot probably, is the robot starts asking Marta, what is this? And when Marta is basically showing a bounding box of a transparent glassware, right? That I show you we worked on that for several years, right? Just trying to find out what is this beaker, right? It says that's the waste beaker, right? So here's the camera, right? So here's the camera, it's looking at the things and asking Marta, let's see if I can raise the volume again.
What will this be used for?
That is the washing beaker.
What will this be used for?
That is the scale.
What will this be used for?
Polishing pad.
What will this be used for?
That is the experiment beaker.
So anyway, you saw that. As I'm talking to you, I'm going to wrap up. I'm going to wrap up and tell you where we are as the video is happening. But this also has a compiler, right? Imagine that you have all your hardware and you want to do things in parallel. So it knows that it can pump while the arm is moving, for example, and things of that sort. That it can steer or do these kinds of things. So it also plans the order of actions in the experiment. It's not pre-programmed. Probably most of you pre-program all your those, do this, do that, yourselves in a script. This was all figured out by the AI, okay? And finally, what's really cute, will appear in the video later, is of course, with the capabilities of ChatGPT-like systems, you can also generate a report, so it generates a report. And you can see in the next slides, which I don't have time to show you, but I could show you the experimental data versus the robotic data. But robotic versus human is about the same. So now you might ask yourself the question, who's happier? So we use the NASA Task Load Index, if you have heard about it. And found out the humans are happier talking to the robot, even if it's annoying, and then going and watching Netflix rather than actually doing all the 40 titrations by hand. But they were faster, actually. The humans were faster than the robot doing this in terms of the robot moves slowly, right? This is accelerated and, you know, but still it was a pretty interesting type of experiment. So now, basically to end, where are we going, right? So we keep working on basic fundamental computer science problems. For example, the pick and place problem is very hard in a messy lab. How many of you guys remember being in a hood? Raise your hand if you have used a hood to do chemistry or biology, right? So now we're looking at a small universal robotics arm that can go inside of a hood to actually operate things inside of a hood. Can you imagine how crowded that environment is, right? So to be able to do that, right, we need to actually have much more data about pick and place. So you have these undergraduates right now using this new Stanford-developed pick and place, like 3D printed robotic arms, that they can use to manipulate and do chemistry and give us the data set that we need, right? Because you need a lot of data set about how to pour things, how to pick things. And this is very good, new modern way of doing these things for computer science, is you have these little arms, and then you have undergrads or people like that doing experiments. So you get a data set that allows you to actually do that. So we're working on that. We're working on more sophisticated ways of doing this. Now we bought our second arm, displacements, and of course, mobile robots come next to get to the point of humanoid robots. But that's where we're at, and maybe it's controversial, but again, I believe the future is going to be robots doing chemistry like the humans and not humans trying to feed their chemistry into these machines. And with that, I'm going to end, and if I have questions, I'm happy to talk to you about them. Thank you.
Thanks, Alain. Very entertaining. That's amazing. Any questions?
Yes.
Ross, hang on. Let me give you the mic.
Hang on. You gotta use the mic, Robert. Can you talk a little bit more about the ROS or the ROS2 program or the robot operating systems that you had on the slide that you're utilizing and how that helps for the integration and maybe tying in with the AI?
That's correct. So we use ROS to control the arm. That's a Franca robot arm, and ROS is a good abstraction to how to control the Franca robot arm. So we talk to ROS from ChemOS, and we send ROS instructions. We also have instrumented some of our chemical equipment as servers in ROS. I think they're called servers, but basically inside of ROS we can also have like balances and things like that. So that's how we kind of under the hood talk to the robot. Yeah. Anyway, happy to introduce you to Kurosh, my property robotics postdoc that will actually answer more deeper questions about ROS. If you have like some technical thing, you will think that as a chemist, I shallowly will get into something I don't know about ROS. But we use ROS as our main and we use PDDL, although we have deprecated it by large language models for the planning. Yeah.
You talked about humans fitting their experiments to these kinds of platforms, and then you said, no, let's have the AI do experiments more like humans. But is there a third option? The AI doing experiments more like AI wants to do them? Are there more efficient ways still?
That's beautiful, of course. So like a dream would be that if we use some generative models to generate the self-driving lab, right? So it would be an interesting idea where the lab itself is generated by the AI, all the components of it. And there are very interesting early versions of that, like my friend Likron in the UK has done this kind of thing that I really like 3D printed reaction where he prints like basically like a chemical engineering kind of workflow thing where like you have two liquids that come in, they get filtered, they get together, and they react, and then separates them somehow as a 3D printed instantiation of his process. So I would imagine that yes, the future could be that a lab builds itself, but I think what we really want to think about is permanence, like renovating a lab for self-driving labs, right? Like right now we're building a new building for them, right? It's $200 million or something, right? So building a new building for it. So I'm thinking not everybody will be able to do this. So that's why I'm thinking for the next maybe 20 years, let's use the labs that are already in Mexico or in Bolivia or whatever, already configured as they are, and have cheap robotic arms use what we have there already. But I think your idea for a future where the lab is conceived by an AI from the scratch, hopefully with cheap parts, it would be fantastic, right? So a robot that builds itself, right? I would love to have a robot that puts all my little wires together in my medusas because we put them by hand, right? So that would be pretty, like, how do you call that TV show that was really cool of these androids? Well, anyway, that would be like this android's vision, right? An android that builds itself. Yeah.
So,
yeah. But that's in the future, perhaps by the time I retire or something. Yeah, I won't care enough about it anymore. Yeah.
Hi, great talk. I'm a postdoc here at UFT. We're talking about self-driving lab, but maybe with this power you could imagine a self-replicating lab in the future, where the lab will print its own components and will...
I think that's what my colleague was more or less asking. Yes, like a lab that prints, or what I was talking about. Yeah, prints or builds its own things. Yeah.
But can like make it better and better and better in each generation.
The lab itself. I like how in this audience people are thinking about the lab. We usually think about the chemicals that the lab makes, making them better and better, or the biology that it's making. You're talking about the lab iterating itself, and that sounds like a cool project. Yeah.
Thank you for a great talk. I have many of the big discoveries just happened during these failures and tedious, boring pipetting and mixing in the lab. It would be great if all of this can be done with a robot or any, that can do an experiment and plan the next and until we find the best point or the optimum point. But don't you think if this might limit the creativity that happens and limit many of the big discoveries that happened before?
No, I think that's question. I have an answer to your question. Okay, so I think of I get your question in a form or another in all my talks. So I have already a pre-canned answer for you. I'm like a ChatGPT. Okay, think about this. Okay, we still have to build a robot. We still have to give it the exploration space that it needs to do, right? So Ilya just submitted a fantastic paper again on how to do sequential dosing into organoids of drugs, right? But by no means the self-driving lab was automated. Like at iteration 3 or 2, I forget which one it was, we had to change the thresholds and continue, right? So the human is involved. But there's a poem by Jorge Luis Borges. How many of you have read Jorge Luis Borges, the Argentinian writer, poet? Anyway, he's like the top, okay? So Borges wrote that poem about chess. Okay, that is talking about the question. It says, who is the god behind the god that moves the player, that moves the piece, that starts this game of dust and agonies? Okay, it's talking about chess, like who's moving the pieces? Well, a human, but who's moving the human? A god? Who's the god behind it? That's my opinion about the self-driving lab. The self-driving lab is like a little mini god or whatever that we're building, but we built it, right? And I am an atheist, so I don't believe God built me. But we are the gods that build the self-driving labs, yeah.
So last question for me. Are you figuring out a way of putting the biology readout back into the chemistry so that the chemistry makes the specific molecule for the biology?
Yes, so I mean, I'm not that biologically oriented, although I collaborate with people that do biology experiments. But the Acceleration Consortium, the thing that I'm directing now, does have labs that are focused more on, I would say, not all the way to biology, but all the way down to like drug discovery and testing of drugs and so on. If you're talking about things like biosynthesis, or I don't know if that's what your question is leading towards, or if my chemistry could help for biology.
No, it's like, say, a drug target, basically, yeah.
Yeah,
Chemistry for
so
drug
yes, I think we are very excited of collaborating more and more with biologists. I think biologists automated their science before us, way before, just because of the fact that biology is very modular. You need to make DNA, RNA, etc. And also you can handle cells extremely well and things like that, right? So I think biology is way more automated than chemistry, right? But I think maybe the biologists can learn from what we're doing here in chemistry primarily, which is these new labs that are pretty bottom up, right? So maybe how can you do things bottom up instead of the traditional 96 well plate? I mean, what I didn't show you, but it's pretty cool, is a computer vision 96 well plate handler with our arm. So we can move the 96 well plate under the arm, and the arm just follows it and dispenses on it. That's very interesting because then you can drop a 96 well plate on the table and tell it fill it. And then you don't have to put it in an XYZ position. So we are doing that so that you are free to now just leave your well plates there, and our robot will figure out how to deal with them with computer vision. Yeah.
Okay,
so that's an interface, yeah.
Let's end it with there. Let's just thank Alan again for taking his time and coming here. Thanks a lot, Alan. And with that, I'd just like to wrap up LRG Toronto 2024. Thanks a lot for all of you coming. Thanks a lot to all the vendors who came out and supported this venue. And we hope to see you all in the future. We're planning on Vancouver for 2025, and there will be a lot of social type events happening in Toronto, Montreal, and Auvergne. So please sign up to the LRIG website. Take a look at us through LinkedIn, where we'll advertise a lot of these things, and hoping we can grow this community. And if you need feedback on automation, just go to the LinkedIn group and make a shout out, and a lot of people will answer your question and help you along. Thanks a lot, everyone. Have a good evening.